Aloha, this is Roger Brewer with the Hawai'i Department of Health. This is a recording of a webinar I gave on November 20th, 2024, titled Field Study of PFAS Associated with Wastewater Treatment Plants, Landfills, and AFFF-release sites in Hawaii. Around 100, 200 people joined in on the call. It's a lot of the people joining in have given us ideas over the past several years on how to assess risks associated with PFAS, how to test for PFAS in the laboratory and collecting samples in the field. So I wouldn't really call this Hawaii's study or even Hawaii's guidance. It's really a combination of lots of ideas from many people. So the question we were asked by our management here in Hawaii, we got a rather late start compared to a lot of other states on the risk posed by PFAS, was, what do we really need to be concerned about in terms of sources of PFAS in Hawaii? So after discussing this issue with a lot of our colleagues in other places, we focused on wastewater treatment plant, effluent, wastewater treatment plant biosolids might be used in compost and such, landfill leachate, and of course fire training areas where AFFF firefighting foam has been used. We went through several iterations of our field study and the investigation. Initially, we were just getting started, so we didn't test for a full suite of PFAS, but as we started to get data and talk to people with other experience in laboratories and such in PFAS, and we added more analysis and came up with different approaches to look at all the data that we're getting. So it's quite an iterative approach between coming up with questions to ask and how to collect samples, what analytical methods to use, what do we do with all the data. Here we'll talk about how we group PFAS into different combinations of compounds to be able to assess cumulative or total PFAS risk. So outline that first off, I want to go over PFAS basics. Again, we covered this in previous webinars, but I think we're getting a little bit better handle on how to present it now. I want to talk about risk-based grouping of PFAS, try to deal with all the complex chemistry and approaches for estimation of total PFAS risk. Then we'll look at some of the data from our field study. And back to the original question, we'll start looking in more detail at relative source strengths, as I call it, of these different potential sources of PFAS here in Hawaii. Then we'll end up with some takeaway points, some thoughts on what we found, and then additional studies. And the key references, the first is the document or field study of PFAS associated with wastewater treatment plants, landfill, leachate, AFFF-release sites in Hawaii. That was posted last week, I think around November 14th, 2024. Pretty quickly after we posted it, I caught an error in one of the figures, so I had to revise that and correct it. So if you look on the second page of the PDF file of this report, then you'll see a list of updates. And I fully expect additional updates in the future. We plan to collect another suite of samples from at least wastewater treatment plants sometime in early 2025, and this report will be amended with the data that we get then. The second report, interim soil and water environmental action levels, EALs, for perfluoroalkyl and polyfluorinated substances, we refer to as PFAS, and we add the S on the end. And that was also updated in November 2024. We'll be looking at that. And again, that document I'm sure will be updated in the future. We also posted toxicity reviews for fluorotelmer alcohols and perfluoroethanoate or trifluoroacetate. We needed these. We started to see these compounds in our sample data. So we needed toxicity factors. There weren't any already published in the US. So we contracted a group of epidemiologists and toxicologists to review toxicity studies and provide toxicity factors for us. So then we've incorporated these compounds into our environmental action levels. And the last document here, technical guidance manual. This is our office's guidance manual on site investigation design and sample collection and such. If you're just getting started in PFAS, then ITRC's 2020 document it's a good place to start. There's a lot of stuff on the web, but they give a good general overview of PFAS. And that document has been updated probably since 2020. So it's a good start. We're going to talk about total oxidizable precursor methods. Here are TOPs, so refer to it. So a number of papers published on this. It was originally developed by a group from UC Berkeley with David Sedlak and his students and others there. They have some papers on it also. I like this paper by Ate et al. It it's a little bit less technical, a little bit easier to follow in the intentions of the method and some of the pros and some of the shortcomings of it. We also have a PFAS webpage, just general information on PFAS and links to our action levels and such. And then another webpage with recorded webinars, which is where you'll find this webinar, at least links to it, along with a lot of other webinars on sampling methods and other topics. We also collected samples of soil around really remote areas of Hawaii. So this would start with this. And we had heard that and read articles about how you find PFAS in glaciers in the Himalayas and such. So it's raining down on us from the atmosphere, from distant sources. So we went out to 9 different remote areas in Hawaii and collected soil samples, had them tested for a full suite of PFAS. Before TOPS analysis, after TOPS analysis, we tested for extractable organic fluorine, total organic fluorine. And we didn't really find anything. In one sample on Oahu that was up in some cliff areas right above the town of Kaneohe, if you know where that is, found some trace levels of perfluorobutanoate. It's barely above detection levels. There were also trace levels of extractable organic fluorine in a couple of the samples, but talking with the laboratory, they didn't really trust the data. It was barely above detection levels. And they're just working on the methods. They're actually retesting those samples for us, and they don't expect to find anything. So good news for people visiting Hawaii or people living here. We're not getting rained down on, apparently, with PFAS from the atmosphere. The chemistry of PFAS can seem really complicated. It is when you start looking at some of the precursor compounds and such being developed, but I like to keep it simple. So we have carboxylates and sulfonates, and especially with respect to assessing of risk, we focus on terminal PFAS compounds. And these are fairly simple compounds referred to as forever chemicals. So we've all heard of butane. So butane is 4 carbons surrounded by hydrogens. So to make perfluorobutanoic acid, we just get rid of the hydrogen compounds, replace them with fluorine, and then on the end of it, put an oxygen and hydroxyl group. So we have perfluorobutanoic acid. If we stuck a sulfur on the end of this instead of a carbon, then we would have perfluorobutane sulfonic acid. Same thing with octane. Take octane, 8 carbons, get rid of the hydrogens, replace them with fluorine, and same group on the end of it, we have perfluorooctanoic acid. If we put a sulfur on the end of it instead of that last carbon, we would have perfluorooctane sulfonic acid, or PFOS. PFOA, PFOS. So it's really quite simple if you just focus on the terminal PFAS compounds. Now, once these compounds get released to the environment, in the presence of water, the hydrogen is going to get stripped off, and we end up with the anionic form. And that's what we focus on in terms of assessing risk and for laboratory analysis. It changes a little bit in the fate and transport of these compounds. So if you look at our guidance, you'll see perfluorobutanoate and perfluorooctanoate. Again, these are the anion forms. That's actually what the laboratory analyzes and reports, or they test for. But in their reports, in the SOP for the laboratory analysis, it recommends they convert it back into the acid form, even though that's not what in the sample to begin with. That's why you see perfluorobutanoic acid and perfluorooctanoic acid and such reported in laboratory reports, even though in reality it's the anion form. So I'm sure you've heard that the short chain compounds now used in different formulations are less toxic than the long chain compounds. So in this diagram, we look at the relative toxicity of the individual PFAS compounds in comparison to perfluorobutanoate, one of the least toxic short chain compounds. So I've set perfluorobutanoate equal to a toxicity of 1 and compared all the other compounds to that, looking at the reference doses for these compounds. So if you look at the bars, the red bars, these are the longer chain compounds. These compounds are more than 100 to even more than 1,000 times more toxic than the butanoate. As you get to the short chain compounds, there's compounds in green, and the ultra short compounds also in blue. See, they're about 10 times more toxic than butanoate, so definitely far less toxic than the long chain compounds. So we got curious, what do we mean by less toxic or lower toxicity. So it compared the reference doses for more other common contaminants to the reference doses for these short chain compounds and the PFAS in general. And probably not, we wouldn't consider them necessarily non-toxic. The least toxic PFAS compound, the perfluoroethanoate trifluoroacetate, it still has about the same toxicity as naphthalene. And you can see the PFBA, the butanoate, has the same toxicity from a non-cancer standpoint as hexavalent chromium, PCE, benzene. You start looking at the other short chain compounds, DDT, TCE, arsenic, even up to PCBs. So these compounds, even though they're short chain, far less toxic than long chain compounds, you still would consider them potentially toxic. In this table, we summarize toxicity factors or toxicity-based action levels for direct exposure to soil, looking at residential exposure, and for tap water or drinking water action levels. So we have 19 terminal PFAS compounds with toxicity factors. You can see that the numbers in yellow here, these are the long chain compounds. You can see they're a lot lower in the action levels than the numbers in green, the action levels in green. The green are the short chain compounds, and the blue are the ultra short compounds. So this is just restating that these longer chain compounds are more toxic than the short chain compounds. So therefore your action levels, screening levels in soil and for drinking water are going to be lower. I've also added in the MCLs, the MCLGs recently promulgated by the US EPA. They're roughly equal to the toxicity-based action levels, just rounded a little bit. Keep an eye on these action levels, especially in tap water, drinking water, for these two ultra short compounds that we have toxicity factors for and action levels. So the toxicity of these, again, much less toxic than the long chain compounds. The perfluoropropanoate, the tap water, drinking water action level comes out to 510 nanograms per liter. It's about the same toxicity again as benzene. Just look at non-cancer risk. Perfluoroethanoate, trifluoroacetate has the same toxicity again as naphthalene, or about 18,000 nanograms per liter or 18 micrograms per liter. Again, about the same as naphthalene from a non-cancer standpoint. We'll be using these later to look at assessment of risk. These are all set to a hazard quotient of 1, which means that these action levels assume that there are no other associated PFAS compounds in your sample. Of course, that's never true. With PFAS, it's usually a pretty complex mixture of compounds. So because we set these to a hazard quotient of 1, it means we have to look at cumulative risk. And that's what we developed an approach to do that. We'll discuss that later. That's the terminal PFAS. Those are easy. We have treat them like any other compound. We have action levels and such for them. It's the precursor compounds that are the next group of compounds we had to deal with. These are terminal PFAS compounds with something else added to it, a functional group added to the end of the compound. Most of these compounds are not reported by the laboratory, and the ones that are reported lack toxicity factors. So that poses a big dilemma when we're trying to assess risk. A good example of this is firefighting foam, AFFF. The main PFAS compound used in firefighting foam after 2005 was 6,2-fluorotellumer thioamidosulfonate. What the 6,2 means, it has those 8 carbons. It was the octane, but in this case, only 6 of the carbons have fluorine attached, and 2 of the carbons don't. And again, then plus there's this big functional group attached to it. We'll look at that again in a minute. Then we have these precursor compounds used in treating textiles and carpet and food containers, at least until recently. One example of these is this 6,2-fluorotellumer alcohol. So again, 8 carbons total, 6 of them have fluorine attached. So it's these short chain PFAS compounds again. So we definitely see that. I think at least the metabolites in some of our samples we'll look at. Then wastewater gets really interesting. If you like chemistry, you go work at a wastewater treatment plant. There's some really interesting chemistry in terms of PFAS. I'm sure lots of other stuff going on. You know, lots of metabolites in the wastewater and this other organic fluorine compounds. We're just really not sure what they are. And then in the biosolids we tested are just full of these odd metabolites, partial breakdown products of these short and long chain PFAS compounds. So we'll be looking at those in more detail. So that was a big dilemma. How do you deal with all these compounds when you're assessing risk and designing cleanup action? It turns out Mother Nature does break down PFAS to some degree. This is a paper on natural oxidation and breakdown of 6,2-fluorotelomere-thiamidosulfonate. It's a compound used in AFFF firefighting foam after 2005. I think they're using a fungus or something to break it down. But the idea is that it mimics what would happen if we ingested this PFAS compound in firefighting foam in our body. What would happen to it? So it would metabolize. Eventually, it would break down into these secondary terminal PFAS compounds. So you can see here that the gradual breakdown of this compound. First, you're developing different suites of metabolites through oxidation. So I circled here where you're adding an oxygen to this functional group on the end of it. Once you get down about halfway through there, you'll see the 6,2-FTS. We definitely see that at sites impacted with modern, post-2005 firefighting foam. We'll look at some data later on. And you see the functional group by now is totally gone. The bugs or fungus or whatever broken it off. But watch what happens next, because in this case we still have 6 of the carbon atoms fluorinated. But the next step, what you'll see this 5,3-fluorotelomere carboxylate start showing up. We see that in quite a lot of samples, not just from the breakdown of this firefighting foam compound, but now only 5 of the carbons have fluorine attached to them. So it struck us, what happened to that other fluorine? And if you look here, just the fluorine the carbon fluorine bond is getting broken. In this case, so you have a fluorine or fluoride ion out there drifting out in the water or something. So that's out in that's what we refer to as PFAS debris. Or the entire end, maybe the entire end of the compound is getting broken off. So you have a carbon with 2 fluorines attached to it. If you go all the way down to where it's broken down now into these forever compounds, the secondary terminal PFAS compounds, you have the pentanoate and the hexanoate. So you have 4 and 5 carbons of these compounds are fluorinated. So more of the fluorine is getting stripped off of this compound, either as fluoride ions or probably also, or you could be also, let the chemist figure this out, 2 carbon molecules with fluorine attached to them. Now they're drifting out there into probably a water solution. So more of this PFAS debris. And then there are other types of these ultra short PFAS compounds that we see in our samples. And that's the curiosity now, these actual breakdown products of these precursor compounds versus originally present in the sample, in the material itself. It may be a little bit of both. So you see the trifluoroacetate, the perfluoroethanoate up there, and also the perfluoropropanoate. It's just basically fluorinated ethane, fluorinated propane that we see in samples. So I'll keep that in mind, because that's another group of PFAS we have to think about. But for now, we know how to use TOPS to deal with these precursor compounds. So if you look at what's originally present in the sample, we have primary terminal PFAS. I just give an example here, perfluoroheptanoate or perfluoroheptanic acid. Then you see the precursor compounds, again in firefighting foam using that same compound. Basically, it's perfluoroheptanoate with a functional group attached to it. That's how you make 6,2-fluorotelomere thiamidosulfonate. What TOPS does, this process, you jack the pH up to, I think, 12, and oxidize the sample. It's going to break off that functional group. And it could even break some of the carbon-carbon bonds, at least, if not some of the fluorine-carbon bonds. I like to think of this as pruning a tree. We've discussed in other webinars, just to keep it simple. So think of the leaves and the small branches. These are the functional groups that are attached to these terminal PFAS compounds. So what TOPS does is it prunes the tree. So it's going to break off these functional groups, and you end up with the building block of that compound, of the terminal PFAS compound. And we can use this to assess risk. So you have your primary terminal PFAS that are present in your sample to begin with. Then you hit it with TOPS, and you break down these precursor compounds. And you end up with another group of secondary terminal PFAS compounds purely associated with the breakdown of precursor compounds. And you'll see in some of the data, it's very typically, you see the butanoate, or you definitely see the pentanoate and the hexanoate, and also probably some ultrashorts getting generated. So now we have three groups of PFAS, and the first two groups are pretty easy to deal with. The primary terminal PFAS, again, originally present in the sample. We just compare the data directly to our action levels. Again, we'll have to look at cumulative risk. We'll talk about how to do that later. And then we have another group, secondary terminal PFAS, generated by TOPS breakdown of precursor compounds that were in the sample. And again, now we have these 19 targeted terminal PFAS, again, direct comparison to action levels to assess risk. But then we have this third group of compounds. Remember we talked about all this PFAS debris that it's predicted you would see in samples, especially in water samples. We see this, and that's identified by testing the sample for total organic fluorine. So the way this is done, and we'll look at an example later, you can calculate how much excess organic fluorine is in the water by taking the measured total organic fluorine, the lab reports, and subtracting the predicted total organic fluorine based on the types and the concentrations of compounds you identified as primary PFAS and as secondary PFAS. Now we run non-target analysis on these samples to try to figure out what's the excess organic fluorine related to, which we see quite a bit of, especially in water samples. And comparing that, also looking at the degradation pathways of the precursor compounds, it suggests that a lot of the excess organic fluorine we see in samples is related to ultra-short compounds in the sample that aren't reported directly by the laboratory. Again, we see this in our sample data. We're actually alerted to this by some of our colleagues in California EPA have been testing thousands or tens of thousands of drinking water samples, and they've been identifying, as I understand it, a lot of excess organic fluorine that can't be explained by laboratory data in the samples. So how do you deal with this excess fluorine, assuming it's related to PFAS compounds? Again, you take your measured total organic fluorine reported by the laboratory, subtract your predicted organic fluorine based on concentrations in the specific chemicals identified as primary PFAS and secondary PFAS. The way we address this in our guidance for assessing potential health risk is we're going to assume that all this excess fluorine is related to the ultra-short compound perfluoropropanoate. We only have toxicity factors for two ultra-short compounds, the propanoate and then the trifluoroacetate. The propanoate is more toxic, if you just remember, has a lower drinking water action level, same thing for soil. So we're going to assume all this excess fluorine, organic fluorine in the water, soil, has the same toxicity as the propanoate, as the mixture. So the way we do this, then we just convert the excess fluorine concentration to equivalent concentration of perfluoropropanoate, and then compare that to our action levels, and we calculate a hazard quotient. And this is done, see in the next box here, it's just kind of basic stoichiometry. You take your reported concentration, or actually your calculated concentration of excess organic fluorine, multiply that by the molecular weight of the propanoate, and divide it by the number of fluorine atoms so that in the compound of propanoate and the atomic mass of fluorine. So this breaks down or boils down to the concentration of excess organic fluorine calculated for a sample times 1.73. So about 70% more than the reported concentration of excess organic fluorine. And that's we refer this as our excess organic fluorine PFAS or excess fluorine PFAS concentration. So now we can compare that directly to our action levels for perfluoropropanoate, same as we do the standard terminal PFAS compounds. You see the action level there for soil and for drinking water. So now we've identified three different groups of PFAS in terms of risk that we can use to assess total PFAS risk. So again, we have our primary terminal PFAS risk plus the secondary terminal PFAS risk, and then add the excess fluorine PFAS risk, and we can estimate total PFAS risk posed by this entire complex mixture of PFAS. Now, we list the laboratory methods here that we use. We dealt mostly with SGS laboratories. So we see for primary terminal PFAS, it's a standard PFAS method there, MLA 110, or if you're using another laboratory, its method, US EPA method 1633 or 537 modified. So 537 modified method has been changed enough to work, at least for our particular work. That's what we mainly focused on if we were testing samples outside of SGS. Again, it's all before TOPS processing. To address the precursor compounds and get our suite of secondary terminal PFAS, we use SGS method MLA 111, or if you're using another laboratory, the same method 1633 or 537 modified after TOPS processing. And for excess fluorine PFAS, then we test for total organic fluorine using SGS method 119. For liquids, it's absorbable organic fluorine. For solids, extractable organic fluorine. So actually, SGS hasn't set up a method to test for absorbable organic fluorine in liquids yet. We weren't using it because we were filtering all our samples, and they said that's an issue with the way they test their samples. So we used urofins to test our samples for absorbable organic fluorine for our liquids. And then the ultra short compounds is SGS method MLA 120. That's just started, I think, this year. Or if you're dealing with another laboratory, just request that they add ultra short compounds to your standard list of PFAS being reported, either pre-TOPS or post-TOPS. It's a different method, so you have to request it specifically. It's basic risk assessment. For the secondary terminal PFAS, again, these are the precursor compounds. How you deal with those, we're going to trim the tree. We're going to hit it with TOPS and break them down to the building blocks, a suite of secondary terminal PFAS compounds. So here you can see the post-TOPS concentration of the individual compounds, along with the primary PFAS concentration pre-TOPS. So to determine the concentration of secondary PFAS, pretty simple. You just subtract the primary PFAS pre-TOPS from your post-TOPS concentration, and that's the concentration of terminal PFAS compounds associated with the breakdown of your precursors. So in case of PFBS now, after TOPS, got a concentration of 8.2 micrograms per kilogram. Compare that to pre-TOPS concentration of 1, so 8.2 minus 1, 7.2. That's the concentration of PFBS associated with the breakdown of the precursor compounds. So we do that for each one of the compounds. You can see the difference there. First, look at the total down at the bottom. The total concentration of PFAS in the biosolids, just based on your primary testing pre-TOPS, 26 micrograms per kilogram. Oxidize it, and the concentration jumps up to 382 micrograms per kilogram. So over an order of magnitude higher. That's because these biosolids are full of precursor compounds. And they're oxidizing, as you can see here on the breakdown, it's mainly into butanoate, pentanoate, hexanoate. So they make up the main contribution. So if you're only testing these biosolids for the standard pre-TOPS list of PFAS, you're missing more than 90% of the total PFAS in the sample, and you could be significantly underestimating risk. But now we've done that, so now we have concentrations of secondary terminal PFAS associated with the precursor compound, so we can assess the risk posed by these. Concentration of perfluorobutysulfonate, 7.2 micrograms per kilogram. Divide that by the soil action level. Here I've rounded them off, which is what we would do, assessing risk. 16,000 micrograms per kilogram. Again, it's a nothing hazard quotient, lots of zeros. So in itself, not a significant risk. Do that for each one of the compounds detected, and then add that up, and you end up with a hazard quotient of 1. Now it's twice as much as their primary terminal PFAS. So your total risk so far now is you add up your hazard quotient for primary PFAS plus your secondary PFAS, you end up with a hazard index of 1.5. So now suddenly your risk is 3 times higher than what we initially thought it was if we'd only looked at the pre-TOPS data. Still not that high, remember, it's just barely over 1. Assessing risk for primary terminal PFAS and even precursors now, we have a good method approach to do that, I think. Fairly simple. So how do we deal with this third group of PFAS compounds, we refer to as excess fluorine PFAS, assessing non-cancer risk? Again, it's the same biosolid sample. So first, look in the yellow column here. Here we've added up the concentrations of primary and secondary PFAS. We're back to our post-TOPS concentration. So you see for PFBS, the butysulfonate, the total concentration 8.2 micrograms per kilogram in that sample. We know the molecular weight of this PFBS, 299. We know it's got 9 fluorines. Using basic stoichiometry, we can actually calculate the proportion of that concentration that would be attributed to organic fluorine versus other stuff in the compound. So in this case, looking at the concentration of PFBS and the molecular weight and the number of fluorines and such, then we can predict that we would have 4.7 micrograms per kilogram of organic fluorine associated with this compound in the sample. That's our predicted concentration of organic fluorine for that compound. Do that for each one of the samples, each one of the compounds, then add it up, and we come up with a total predicted organic fluorine concentration for this sample of 265 micrograms per kilogram. We had the lab test it for actual concentration of total organic fluorine, and we see this over and over in our, whether it's a water sample or the biosolids or soil sample, and they report a concentration of total organic fluorine of 842 micrograms per kilogram, so way above what we would have predicted. So you subtract the measured from the predicted. This is what we refer to as excess organic fluorine in the sample, 577 micrograms per kilogram. So how do we address this again? If you recall, we're going to assume that all this mixture here has the same toxicity as perfluoropropanoate. And looking up on the left, the boxes here. So we take that concentration of excess organic fluorine, multiply it by 1.73 to calculate an equivalent concentration of propanoate, in this case 998 micrograms per kilogram. And we have a concentration of excess fluorine PFAS that we can use to assess risk. So looking at the next box, and to calculate a hazard quotient for this, we just treat it the same way we treat any other compound or our primary or secondary PFAS. We just divide it by the action level. In this case, we're looking at solids. We compare it to our soil action level. We're using residential again, so 998 micrograms per kilogram divided by 5,100 micrograms per kilogram, then comes out to a hazard quotient of 0.2 for these excess fluorine PFAS. So now we have a method to look at total PFAS risk for this sample. Again, we just sum the risk, the hazard index, calculated for primary terminal PFAS and secondary terminal PFAS, plus the hazard quotient. In this case, estimated for the excess fluorine PFAS, and that's our total PFAS risk. So 0.5 plus 1.0 plus 0.2 equals a total hazard index of 1.7. We round it up to a single digit, so it's 2. So that's our total PFAS risk here. Anything over 1, means you want to look at it in a little more detail. You know, hazard index of 2 from a risk standpoint, not a big deal. There's a lot of big safety margin built into these toxicity factors and methods to assess risk anyway. So it wouldn't cause risk assessors to jump that much. It is over 1. Let's take a closer look at actual exposure and some of the assumptions in the models used. And again, you see that in the bottom box here, hazard index less than 1, no anticipated adverse health risk. Over 1, just review your actual exposure, your risk model assumptions, toxicity studies, and such. So that can get kind of lengthy. I think the approach is fairly straightforward now. At least risk assessors should be familiar with this approach. But to keep this simple to use, then we prepared a spreadsheet in Excel for calculating total PFAS risk, the hazard indices and quotients associated with each one of these 3 groups of PFAS. It's I think quite easy to use on the first page, first worksheet. That's what you see here. The first step is to input basic sample information. You can see here it's an example for wastewater treatment plant number 6, the biosolids. I put in a date there. You put any other information you want. Then the next step, you select whether it's a solid or liquid media. If you select solid, then it's going to compare your input data to action levels for soil. If you selected liquid, it compare it to our toxicity-based action levels for tap water or drinking water. Third step only applies to soil. Then you would select the land use, either unrestricted, residential land use, schools and such, or you can select commercial, industrial, or even construction worker. And this just refines the set of soil-based action levels that you'll be comparing your data to. Step 4, optional. It's kind of a risk assessor's only little box we stuck in there. If you don't like our approach of assuming all this excess organic fluorine associated with the same toxicity as propanoate, you can input your own toxicity factors and your own risk-based action levels, either soil or tap water in there, and you would just justify that in a risk assessment. Again, not too many people will be using that, but we wanted to add it in just for future use. That's the first page. Pretty simple. Second worksheet, you just input your sample data. So at the top, you're inputting your total organic fluorine. In this case, it was for soil, so you see the units up there, micrograms per kilogram. If you'd selected liquid on the first page, the units would change to nanograms per liter. Keep in mind these is micrograms per kilogram. Laboratories often report milligrams. You have to convert it. The second box there, where you see 1.73, that's the default mass adjustment factor for converting all the excess organic fluorine into an equivalent concentration of propanoate. Most of the time, you're not going to be bothering with that unless you selected some other alternative surrogates for your toxicity. I think it actually would automatically convert anyway now, the way it's set up. So you input that, total organic fluorine, and then the last step, you just input your pre-tox, post-tox data in the first column and the second column, as you did here, just cut and paste it in. And then the spreadsheet automatically sums up your pre-tops, post-tops data. You can see again here is a big jump after you oxidize it. That's all those precursors, the metabolites, and the biosolids coming out. Once you do this, there's another set of worksheets, which you can look at. You don't have to, but it's doing all this background work. It's calculating excess organic fluorine in the sample based on your total organic fluorine measured versus what it would be predicted based on your input data. It's coming up with the secondary terminal PFAS concentrations, and it's comparing your primary and secondary terminal PFAS to your action levels and calculated hazard quotients and such. So take a look at that if you want. Otherwise, just go back to the first page and look down at the bottom again, and now all the concentrations for each one of these 3 groups of PFAS shows up, and it sums it up there together. So again, look at the sum for this particular sample of biosolids. If you only tested with the standard PFAS reporting, then without tops or anything, then you would assume the total concentration of PFAS is 26 micrograms per kilogram. Run tops on it and look at your total organic fluorine, and actually your total PFAS is probably over 1,000 micrograms per kilogram. So you'll miss most of it if you didn't run tops in total organic fluorine. And you can see the breakdown here for this particular sample. Most of the PFAS compounds in the sample related to this excess organic fluorine, which are non-targeted analysis and published research suggests a lot of that's probably associated with ultra-short compounds in the sample. Look at risk though, and it's a different story. Most of the risks posed by this suite of compounds in the sample of biosolids actually posed by the secondary terminal PFAS, almost 60% of your total risk, your total hazard index. And again, that's from the breakdown of the precursor compounds. If you didn't run TOPS on the sample, you'd be missing most of the risk posed by these samples. One thing before I forget, if I forget later on to mention it, all these sites we look at, and we're calculating risk, and we'll look at some example risk calculations later. Really none of these sites are being used as playgrounds or anything. There's no exposure. There's really no significant impacts, or there are no significant impacts to drinking water resources posed by the sites that we investigated. Most of them are located over in coastal areas, not over drinking water. So it's all hypothetical. That's discussed in our report. This is just meant to look at the relative source strengths and relative risks posed by different sources. So that's how you calculate total PFAS risk. So now let's finally get to our field study data, looking at data for wastewater treatment plants, landfills, AFFF-release sites. This is just a quick summary of how we collected the samples and how we did laboratory analysis. So our wastewater treatment plant samples, Influent and Effluent, they actually collected samples for us using a 24-hour composite sampler. So the idea was to get a quick snapshot of the, sort of a random weekday in the life of a domestic municipal wastewater treatment plant. For the biosolids, we targeted at least a volume of biosolids that roughly approximated how much biosolids will be generated during a specific day at this wastewater treatment plant. Now, these weren't the same biosolids associated with the samples of Influent and Effluent that we tested, obviously, but it gives us some general idea. The biosolid samples, same with AFFF contaminated soils, we collected 30 to 50 increment, multi-increment samples, so no grab samples. I'll rant about that. Next slide. Then you see another summary of the sample analysis. We already talked about that. We got samples from 6 wastewater treatment plants. These are all domestic wastewater treatment plants. They're not attached to any industrial facilities as such. So most of the, all the influence should be coming from residences or maybe some commercial businesses. And then we have samples of leachate from 5 landfills, and we got samples of soil from 2 AFFF-release sites and samples of groundwater from 1 of the release sites. Now, if you work in Hawaii, then you're familiar with us in our multi-increment sample approach for collecting soil samples and samples of any solids. We haven't accepted the use of data for grab samples, discrete samples, for 15 years now. We've been studying this for 20 years. We have a number of publications and such on the reliability or kind of unreliability, uncertainty of grab sample, discrete sample data posted to our webpage. You can check out the webinars there. This is the fire training area at the 2nd AFFF-release site. There's a grassy area beside the paved area where the fire training area is. So we just assume this is a hypothetical exposure area. Maybe workers at the airport were going out here and hanging out in the grass during lunch or something every day. They really aren't. They might go out there on occasion once or twice a month. But the way we collected this in, we focused on the top few inches of soil. It's pretty gravelly. It's actually hard to get a sample. So we collected small amounts of soil from 45 points throughout this area. Each small mass is referred to as an increment. And then we combine all this together in a single sample. We call it our multi-increment sample. So we're kind of simulating what if we just scraped off the top 4 inches of soil at this site and sent that all to the laboratory to test a single sample. Of course, we can't do that, so we got to represent a sample. Lots of science behind that. Another whole field of science, environmental industry is just catching up to, called Pierre-G's theory of sampling on how to do this. Each sample weighed about 2 kilograms. We actually did it 3 times from independent increment spots within here, and then we compare the data for each sample to test the overall precision of the sampling method used, the analytical method used. Then when the laboratory gets this sample, they don't just open up the Ziploc bag, which we use, and take out a random pinch and test it. They have to air dry the sample, sieve it down to our targeted particle size, which for direct exposure, we use less than 2 millimeter particle size. Then they use sectoral splitter to get a subsample of that, or they spread it out into a thin layer cake. And actually, before they do that, they need to re-wet the sample to keep the fines from settling out. So it's very particular how you do this. And then they'll collect a subsample from that in the same systematic random approach we use to collect the sample in the field. And that way, the subsample laboratory test is representing the sample collected. The sample collected is representing the targeted area. Laboratory has tested minimum 10 grams. There's a lot of science and statistics behind this also. That's a minimum mass of a subsample you need from less than 2 millimeter particles to get a representative subsample of the particles. So that's, I always say every revolution needs a slogan. So nobody's using this one now. We'll use it, stop the grab. So we really have to stop this grab sampling in the environment industry. You're collecting discrete samples and testing them. Probably the sample data for soil samples test for PFAS, I would say some of the most unreliable data in terms of being represented the sample collected. At least in the approach used, the laboratory protocols only call for testing half a gram, a tiny pinch of the sample. And it's physically impossible to represent a sample of soil by only collecting a random half gram mass of it. You really need to do at least 10. And in the field, of course, move your sample point over 6 inches or whatever, and you can get a totally different number. Same thing, testing random half gram plucks of the soil from a jar, you'll get different numbers. How bad is it for PFAS? It may not be that bad. Maybe you're only off by a factor of 2 or 3, probably less order of magnitude total for each point, but that can cause a lot of problems in the field and also assessing risk. So enough of sampling. Let's look at some of the data. This is the wastewater treatment plant number 5, Influent, Effluent, and the Biosolids. Looking at Influent first, this is pre-TOPS total concentration of PFAS. These just the terminal PFAS reported by laboratory, 64 nanograms per liter. That's one point that's really low in our domestic wastewater treatment plants, Influent and Effluent. Even when you're looking at total organic fluorine, pretty low concentrations. This particular plant, the main compound reported was 5,3-fluorotelmer carboxylate, 40% of the total of the PFAS, individual PFAS reported. Now, this wastewater treatment plant doesn't get any leachate from landfills. So this is all coming from domestic use or maybe some commercial use. The second most common compound, the perfluorohexanoate, 25%. That shows up quite a bit, and you see the mix of others. Again, we didn't get total organic fluorine for this sample. This is our first round of field work. We didn't really know much about total organic fluorine yet. We did test some of these with TOPS analysis. You don't see a big change in the concentration. A lot of times it goes down. So what does that tell us? When you oxidize a sample, process it with TOPS, and the concentration go down, well, in that case, it means that you caught most of the precursor compounds were in that sample. In this case, the total PFAS includes this fluorotelmer carboxylate. So you're catching most of what's there. We see that pretty routinely with water samples. The TOPS didn't make a big difference. If anything, the concentration goes down because you're stripping off that functional groups and you have less mass in the post-TOPS analysis. Look at the effluent, though, and all that fluorotelmer carboxylate got oxidized as it went through the wastewater treatment plant, just like you'd expect a good wastewater treatment plant to do. And now it's been oxidized into the pentanoate. You see there in blue. You see the hexanoate, some others. And of course, the more you look, the more you find. There's some 6,2-fluorotelmer sulfonate showed up in the sample. It's usually from maybe AFFF. Who knows where that came from? So they're pretty complex mixtures. You see the concentration of total PFAS in the effluent was a little bit less than the influent. That could just be variability. It's what my initial guess would be in wastewater. You're looking at nanograms per liter, remember. So not a big deal, not much to think about. Look at the biosolids. Pre-TOPS total concentration of reported PFAS, 181 micrograms per kilogram. We had the laboratory test for lots of precursor compounds, everything they could. And one thing we see in the biosolids, they're just sponges, especially for these longer chains. You see that O in the me-phos and in me-phos. That O means octane, so 8 carbons there. So you see a lot of these, longer, shorter chain compounds and long chain compounds in the biosolids, just sponges for this stuff. Oxidize the sample though, and look what happens. The concentration more than triples, up to 680 micrograms per kilogram. And you see these precursor compounds are breaking down into the standard suite, the butanoate, the pentanoate, hexanoate. But it tripled, so what that tells you is, even though we found a lot of precursor compounds even without TOPS, there's still a lot of other stuff in that sample. That's why it's important to run TOPS on your, especially samples of soil and biosolids, to find out what's in there and fully assess risk. We didn't get any total organic carbon or fluorine data for the biosolid samples either, or no non-target analysis. but still very interesting. This is the second wastewater treatment plant, number 6. This one is the highest concentration of PFAS we found in domestic wastewater influent, 250 nanograms per liter, still really tiny, way less than a microgram part per billion. In this case, this particular wastewater treatment plant, they got a lot of their influent, we think, from hotels. It's near a resort, not as much domestic residential use around there. So our guess is a lot of these extra PFAS, again, really not that much, but are probably coming from washing of tablecloths and things that are treated with PFAS for stain resistance, water resistance, maybe bed linens, carpets, and other stuff. In this case, we did test for total organic fluorine, absorbable organic fluorine, 2,000 nanograms per liter. It's almost 10 times higher than just an organic fluorine itself from the total PFAS. So you convert this to excess fluorine PFAS, and you're over 3,000 nanograms per liter. So it's by far, that's the main component of organic fluorine-related, PFAS-related compounds in the influent. Didn't see any precursors here. It's all, even in the influent, it's all the pentanoate. Look at the effluent, about the same as the influent. Concentration of PFAS in the effluent here was higher than the influent. This could just be variability, or it could be that a lot of precursor compounds are initially bound up in the suspended biosolids in the influent are getting broken down into more ultra-short compounds, and that's coming out in the effluent. That's why the concentration of total PFAS in the effluent increase. Now we hit that with TOPS. Again, you don't see a big increase in the total concentration of PFAS after oxidizing it. I think it actually went down again in this sample. That's because there's just not that many precursors left by the time it gets in the influent. Absorbable organic fluorine concentration, again, a lot higher than even the concentration in the effluent, even if we hit with TOPS. So most of the PFAS organic fluorine-related compounds in the effluent, again, is related to these excess fluorine compounds. Biosolids, a similar story. Total PFAS pre-TOPS, 59 micrograms per kilogram. Post-TOPS, 409 micrograms per kilogram, almost 10 times higher. So again, that just means there's a lot of precursor compounds bound up in these biosolids that aren't getting reported in our data unless you hit it with TOPS, oxidize it. So you see that it's more than 2 times, a lot higher than that actually. We did run non-target analysis for both I think it was the influent and the biosolid samples, and they were reporting what they could report, mostly ultra-short compounds. There could be other things in there that didn't get broken down, even in the TOPS, maybe PFAS compounds with fluorine bases or chlorine or something else. But the assumption right now is it's mostly ultra-short compounds. Again, a lot of extra organic fluorine in the sample and be predicted based even after TOPS analysis. And again, that's we're assuming is associated with ultra-short compounds. That was the wastewater treatment plants. Next for landfill leachate. Similar. It looks a lot like a wastewater treatment plant effluent, but a lot higher. It's about a total concentration of PFAS in these samples, about 100 times higher than what we saw in wastewater treatment plants. So a lot more toxic in terms of both concentration and even the suite of PFAS present. You see a lot of 5,3-fluorotelomere carboxylate, which some of our colleagues on the mainland were alerting us to. Said you're going to find that, and sure enough, we did. It's the main PFAS compound, at least reported PFAS, in the leachate. It's not getting oxidized. It's just sitting there fermenting in the leachate. Look at the total organic fluorine reported, 83,000 nanograms per liter in this one cell. These 2 cells on the left are municipal garbage. So a lot of additional organic fluorine compounds in this leachate. Again, we're assuming it's related to ultra-short compounds. We don't have any NTA data yet for it, but that's what the assumption is, and talking with some other people. That's the main component of landfill leachate also. This third box over here by itself, you don't see that much 5,3-fluorotelomere carboxylate there, only 10%. That's actually a cell that's filled with ash from a municipal waste to energy facility. It's burning municipal waste again. So, any precursor compounds or most of the precursor compounds are getting oxidized and broken down before it went into the as it got incinerated and the ash got dumped into the landfill. Total PFAS concentration is still lower in this leachate, and there's still, again, as much organic fluorine in the water. So there's still a lot of precursor compounds in this cell, even though it's already ash. Also, again, same thing we saw in wastewater treatment plants. Their post-TOPS data is pretty similar to pre-TOPS data, which means the main precursor that you're going to find in this leachate that we're finding is this 5,3-fluorotellular carboxylate. Moving on to the AFFF-release sites. This is the fuel storage area. The firefighting system had a leak of about 1,000 gallons of modern AFFF, which means anything AFFF past year 20. Oh, this was released in 2022. So anything past the year 2005, remember, doesn't have PFOS in anymore. It's got this short chain precursor compound, this 6,2-fluorotellular thiametosulfonate. Well, this was a great learning lesson for us. This is back in our first phase of investigation. They real quickly, they got out, dug up obviously contaminated soil. It looked clean, what they dug up. They were out there within a days after it was released. We collected our own confirmation sample, got a good 45 multi-increment sample from the bottom of the excavation, had it tested, pre-TOPS, total concentration of PFAS, 144 micrograms per kilogram. The main PFAS compound was this 6,2-fluorotellular sulfonate, again, which that was expected because we knew this was the modern AFFF. Concentration is still pretty low, and we got toxicity factors for this specific compound. It wasn't that big of an issue. This all come out industrial area anyway. I was talking with the laboratory chemist about this particular sample, saying, oh, they did a good job cleaning up. Didn't find much. He said, yeah, but you missed most of what's there. And that's when I learned all about TOPS. And so we had him oxidize the sample or process it using TOPS. And look what happened. The total concentration of PFAS jumped from 144 micrograms per kilogram to 23,000, almost 24,000 micrograms per kilogram. Well, no big surprise, because that's pre-TOPS. They weren't testing for the right PFAS compound. After you oxidize it, then these secondary terminal PFAS associated with that precursor compound they've been using in AFFF since 2005 show up. That tells us what's really there. Breaking down to that same suite, the butanoate, pentanoate, hexanoate, which is what you would see even in natural breakdown of this, or maybe it's metabolized in your body. It's really highlighted the importance of testing, especially soil samples using these TOPS, which is what we did in our subsequent phases. This is the second site, AFFF-release site, after we had gotten some education from our first round of sampling, talked to lots of people. So again, here's the grassy area highlighted in red here, where I showed you how we collected multi-increment samples and such. So we tested it, and pre-TOPS concentration comes out to 878 micrograms per kilogram. But look at the main PFAS compound. Now it's PFOS, not like the first site we tested. So that throws up a hint. Pretty quickly, this is probably the older AFFF. And at this site, is used since 1977. They lined the area in 1997 for our training area, including the pit, and then kept using it. It's still in use now. But what this tells us, the dominant compound in our sample PFOS, is that's probably an older release. That's from the pre-2005 release, at least, of AFFF in the air when they were washing off the either paved area before they paved it. And we oxidized the sample, and sure enough, it only goes up about 40%, which is pretty minor compared to the first area we looked at, which went up 100-fold or 1,000-fold. Now the total concentration of PFAS post-TOPS, 1,051 micrograms per kilogram. Not that big of a difference. So the main concern here, again, this is an older AFFF-release site. It's these primary terminal PFAS compounds originally present in the sample. But running pre-TOPS and post-TOPS tells us a lot about, from a forensic standpoint, when was this released, who released it, and what kind of AFFF it related to. This is the second fire training area. I mean, the same fire training area again, but in this case, looking at impacted groundwater. Another interesting story, testing for all three suites of PFAS, these primary terminal PFAS, secondary terminal PFAS, and your excess organic fluorine, right in the source area. See the burn spot there in the middle of the red highlight here. You see this 6.2 FTS highlighted in pink. So you see that in your groundwater. Well, you know that's probably, that is going to be related to releases of post-2005 AFFF. So what does that tell you? One thing it tells you is your liner that you put under the fire training pit has probably got cracks in it now. But you still see a lot of the PFOS there. It's actually the dominant PFAS compound in the groundwater. So that's related to the older PFAS that was used there pre-2005. Look at the concentration here. I had to switch to micrograms per liter instead of nanograms because the numbers are so big. So we have ppm level total PFAS in the groundwater right under the pit. Again, this is all pre-tops. If you hit this with tops, you're not going to see a big jump. You'll actually see a decrease, I think, in total PFAS because you're breaking down, you're stripping that functional group off the 6.2 FTS so you get less mass. But again, the total organic fluorine, AOF, absorbable organic fluorine, twice as much as total PFAS. You convert that to an ultra short equivalent concentration, and that's still these ultra short or excess organic fluorine related PFAS are making up most of the PFAS related compounds in the groundwater in the source area. Looking at downgrading areas, here the main compound in the downgrading areas, main PFAS, concentration is dropping off pretty rapidly. It's PFOS. What does that tell you? Well, down gradient, it's mostly this older plume from the use of the older PFAS before they lined it and such. You see a little bit of hexanoate, pentanoate. So what does that tell us? That's probably the breakdown products from the release of this, the later precursor-based PFAS. Again, we talked to some microbiologists. They were saying as soon as these PFAS precursor compounds get in the groundwater, the bacteria break up down into the terminal endpoint compounds pretty quickly. They strip off those functional groups. So you get some good forensics information looking at this. You get to the perimeter. If you look in the upper right here, the total PFAS, you don't see in the PFOS, you see that hexasulfonate, which talking to some colleagues on the mainland, they see a lot of hexasulfonate in the water samples. It seems to be a lot more mobile than would be predicted of otherwise. It's just diffusing out along with these shorter chain compounds away from the main plume here. And where the butasulfonate come from, I don't know, breakdown product or something. Now, the chemistry again is complex, but really interesting. We don't have any NTA data yet for this. So that's the sample data we found. Back to our original question, what do we need to worry about most? Well, the last thing we want to look at is the relative PFAS makeup and the hypothetical risk. Again, none of the sites we look at really pose a significant risk. It's not impacting drinking water. Nobody's playing in the soil, the biosolids. So let's look at that between the effluent, biosolids, landfill leachate, and fire training areas. So this diagram here, we compare the relative PFAS group makeup of those three different PFAS groups for effluent, landfill leachate, AFFF-impacted groundwater. So again, the green bars here for each one of these different scenarios, those are the excess fluorine PFAS. Every water sample we tested, if just looking at the total organic fluorine data, the main PFAS related compound in the water are these excess organic fluorine PFAS, probably ultra shorts, based on the way we're looking at it now. In the wastewater treatment plant effluent, they make up 98% or so of the total PFAS in the water. Just smaller amount of these primary PFAS compounds, the hexanoates, mostly carboxylates. Landfill leachate, same story. About 70% of the total PFAS mass are these excess fluorine PFAS. Again, assuming they're ultra short compounds. They could be other compounds, phosphate-based PFAS as such didn't break down, but we'll assume they're ultra shorts. That's what the story tells us so far. But still a pretty high proportion of primary PFAS compounds, PFOS, PFOA, hexanoate, and such. Not much in the way of secondary PFAS if you oxidize a sample. That's because they're already getting broken down into landfill leachate. And then in the AFFF-impacted groundwater, same thing, the main by mass proportion, whatever, the main suite of PFAS compounds. And this is right in the source areas, mostly the excess fluorine PFAS, all these apparent ultra short compounds. And then in the source area itself, If you oxidize a sample, you'll get a decent hit of secondary PFAS because we had the 6,2 FTS there. That's going to get broken down into the pentanoate, butanoate, hexanoate, and such. And also that 30% of the PFAS in this particular sample in the source area were the primary PFAS, your standard PFOS, hexanoate, pentanoate, and such. That's just the makeup. What about risk? That's the focus. It's a little bit different story. When the wastewater treatment plant effluent is just packed full of these apparent ultra-short compounds associated with excess fluorine PFAS, assuming those have the same toxicity as the propanoate, which toxicologists I talk to know a lot about PFAS, that's being appropriately conservative. But that's what's driving risk in the wastewater treatment plant effluent from a drinking water standpoint. Of course, no one's drinking the effluent. It's just looking at relative source strength. And then the primary PFAS, mostly the short chain compounds, they're the secondary drivers to risk. Landfill leachate, even though the excess fluorine PFAS make up most of what's there, then it's still the primary PFAS, some PFOS, PFOA, and hexanoates and pentanoates and such. They're a smaller proportion, but because they're more toxic, they're driving risk in the landfill leachate. Again, that's just compared to drinking water action levels. How about the AFFF-impacted groundwater? In this case, again, we know there was a mix of older AFFF, modern AFFF spilled at the site, but either way, the precursor compounds are getting broken down pretty quickly in the groundwater, even under the source area. Definitely as you move away from the source area, you don't see any more of the precursor compounds. So the main risk posed by AFFF-impacted groundwater are these primary PFAS. That's a good story. If you're only focusing on these primary PFAS initially reported by the laboratory for plumes, at least near the source area, you're catching most of the risk. The question is, once you get away from the source area, if these apparent ultra-short compounds are all that's left, they've kind of raced away at the leading edge of the plume, they could be driving risk. So we'll look at that again in a second. Those are all the water samples. How about the biosolids and compost, AFFF-impacted soil? Again, the main component of the biosolids and the compost are these excess fluorine PFAS, whatever that is. So my chemist colleagues take a look at that in more detail and let me know what's actually there. What is all this excess fluorine there? And we can adjust how we assess the risk from it. But in the biosolids themselves, that's the main group of PFAS compounds. Again, a lot of precursor compounds, a really interesting complex mix of metabolite, shorter and longer chain compounds there. And then not much in the way of primary PFAS we see in the biosolids, your PFOS, PFOA, hexanoate, and such. It's all these secondary metabolites. In the compost, we see, again, the main group of PFAS compounds is excess fluorine PFAS, all these apparent ultra-short compounds. Not a big surprise. If you remember in the effluent, it's mostly these ultra-short compounds. Well, biosolids, the samples we collected were 85% water, where that water is effluent. It's not a big surprise. There would be a lot of ultra-short compounds left in the biosolids in the compost, even after you cure it. And in the compost too, there's still a fair amount of, the next group would be the primary PFAS compounds, your hexanoates and pentanoates and such, or PFOS, PFOA if they happen to be there in the biosolids. And there's not much left in the way of secondary PFAS. These are precursor compounds. They've mostly been broken down in the compost by now into your primary PFAS. AFFF-impacted soil, we talked about that. Older AFFF, PFOS base, your main group of compounds are going to be PFOS, maybe some hexasulfonate. In our case, we saw some precursor compounds in the older AFFF. It could have been some of the newer AFFF that made its way out there, but hexanoates, pentanoates, and such. And a small amount of excess fluorine PFAS in the AFFF-impacted soil. Once you get to the modern AFFF post-2005, with no big surprise, you don't see any of the primary PFAS compounds. It's all the secondary PFAS compounds. It's mostly 6,2 FTS, at least in the soil samples we tested. And of course, they're going to get broken down into secondary PFAS compounds, the precursor compounds you see here. We didn't test for total organic fluorine in the sample. I wouldn't expect to see much. Of course, you're not going to see a lot of pre-tops terminal PFAS like PFOS and such in the AFFF impacted soil if it's post-2005 because they weren't in the AFFF to begin with. So that's the makeup. Let's look at risk. The biosolids, again, they're all dominated by these excess fluorine PFAS, but they're not that toxic, and there's enough precursor compounds in the sample. That's what's driving risk in the biosolids. So this really emphasizes why you need to process samples of even compost or soil really made with biosolids using tops, because you can identify all these secondary PFAS in the sample, especially if you're just looking at the biosolids themselves, I would say. The second group driving risk would be these primary PFAS compounds, mostly just short chain compounds. And the excess fluorine PFAS, again, they're the dominant group of PFAS in biosolids, but they don't really drive risk that much. If you look at the compost, again, most of your precursor compounds have oxidized now, so if you're testing compost, it's going to be your primary PFAS initially reported without tops that are driving risk in that. And then your secondary PFAS and excess fluorine PFAS, yeah, there's some of the excess fluorine PFAS there, but they're not really driving risk. It's just primary PFAS compounds. So if you're not hitting compost with tops, you might not be missing that much in terms of risk, but it's still useful to do. AFFF contaminated soil, in the older AFFF, it's PFOS based, and no big surprise, your primary PFAS is what's driving risk. A little bit of risk from secondary PFAS in the older AFFF. It could be there. So it's worth looking at. Modern AFFF post-2005 is a great seared up document, by the way. 2017 talks about formulations of AFFF. It's referenced in our guidance. But again, in post-2005 AFFF, it's these precursor compounds that we assess after tops and identify these secondary PFAS. That's what drives risk. So any post-2005 release of AFFF, if you're not hitting with tops, then you're probably missing most of the risks, or you are missing most of the risks posed by that contaminated soil. So I was looking at makeup versus risk. Now we're getting down to the bottom line. What was our first question we started off with quite a while back now? So in terms of what do we really need to be concerned about? So look at what I refer to as relative source strength in terms of hypothetical risk, at least for the sites we looked at. Look at non-cancer hazard. So this is just comparing the hazard index, total PFAS risk hazard indices calculated for effluent landfills and AFFF-impacted groundwater. So you see in the green here for the effluent, what's the bottom line? What's our total risk here? Even look at all three groups of compounds, we're still looking at total hazard indices, of less than 10. And really less than 10 in terms of risk isn't that high, what most risk assessors I think would look at. And again, we're assuming, remember the main risk here is posed by these excess organic fluorine related compounds, which we're assuming are as toxic as propanoate, the most toxic ultra short compound we know about. So we're probably being conservative here, or we are being conservative. So the risk posed by PFAS and wastewater treatment plant effluent in terms of impacts to drinking water resources is relatively low. Compare that to landfills. So you have a leaky landfill. Leachate, a lot more PFAS and more of the more toxic shorter chain and some long chain PFAS compounds in landfill leachate. So now your hazard quotient's way above 10. That would definitely get the eyes of a risk assessor, even above 100 in some. One thing I forgot to mention too, we did get landfill leachate samples from a construction debris, a dedicated landfill. Similar concentrations, a little bit higher risk, and there are a lot of sulfonated compounds in the leachate from construction debris landfill. So people who are familiar with that type of debris, I'm sure could explain that. Maybe treated concrete and such. But very high risk. So landfills definitely pose a much higher concern than wastewater treatment plant effluent. The AFFF-impacted groundwater here, and the hazard indices here, it's in logarithmic scale. They're hitting 100,000. Anything over 1 is something to look at. Anything over 10, definitely get concerned about. And over 100, you definitely have a problem. Now we're talking about 100,000. So luckily here in Hawaii, we haven't found, we're not going to find any, I think, AFFF sites of AFFF-impacted groundwater from firefighting sites, especially where the groundwater is really shallow within a few feet. That's where you get really toxic groundwater. So if you have a municipal well near one of these sites, which a lot of our colleagues on the main have been telling us about, you can get really high concentrations of PFAS in your municipal well with really high toxicity. So that's our main concern for impacted water. For the solids, biosolids versus compost versus AFFF impacted soil, really biosolids and compost, the good news here, what we've seen pretty consistently between sites, again, just looking at domestic wastewater treatment plants, just residential and your commercial, really low potential health risk, even though it's really interesting chemistry there, but the health risk target calculated hazard indices, usually 2 or 3. Even for the compost, I think it's actually, it'll round off to 0. So not a big problem. With respect to PFAS, if young children or pregnant mothers being exposed to biosolids emitted soils and such on a daily basis, just not much there to begin with, and what is there is pretty low toxicity. The bigger problem is AFFF impacted soils. The samples we collected were all away from the original spill site, so that it had been remediated partially. So the calculated hazard indices are not that high. For the first site, they're less than 10, but that was after they excavated all the really nasty stuff. So if you're looking at fresh releases, that should say AFFF site number 2 in the last slide there, DU5F. That was the second site we collected samples from. This was at the margins of the fire training area, probably a good 100 feet or more away from where I actually used the firefighting foam. Hazard index of over 30, approaching 40. That's pretty high, but I'm sure when they start digging underneath the pavement there, and they're actually going to excavate all the really heavily impacted soil, there you're going to be looking at hazard indices way above 100. So AFFF impacted soil poses a much higher potential risk concern than just biosolids. That's the summary, some takeaway points to finish up here. Just in general, again, it's based on the sample we collected at our sites. But testing of initial samples for a full suite of PFAS, one thing we realized is really important, at least in initial stages of investigation, so you can understand the full PFAS makeup of the impacts and get a more reliable estimated total PFAS risk. So you test the initial samples using pre-tops, post-tops analysis to identify primary terminal PFAS, secondary terminal PFAS, get total organic fluorine data so you can identify excess fluorine PFAS. TOPS data, especially important for soil biosolid samples. Maybe total organic fluorine not as much, but it's useful for forensics and such. Water samples, especially important to get total organic fluorine data. TOPS data, not as important, but again, it's very useful for forensics and understanding what's going on at the site in terms of chemistry. So you could start backing off on your analysis after your initial suite of data once you identify what your main group of risk drivers are, whether it's the pre-tops or post-tops, primary, secondary PFAS, your total organic fluorine. Overall, though, we need to get a better understanding of all this excess organic fluorine in the water. Again, we're assuming it's mostly ultra shorts for various reasons, but chemists have fun with that and let us know what you find. Non-target analysis, it's useful, at least in part, for identification of what all this excess organic fluorine is in a sample, what you're missing in your regular analysis. So it's very useful to get that data, at least on a few samples. Expensive, but important, I think. One thing some chemists and some remediation people point out is these ultra-short compounds. For one thing, they might be going straight through your absorbable organic fluorine test methods. You're not capturing that in your total organic fluorine data, something like trifluoroacetate. So we actually might be under-predicting how much excess organic fluorine is in the water because the absorbable organic fluorine test uses like a carbon sorptive, with some of the ultra-shorts might be going straight through it. So we need to think about that more. PFAS and wastewater treatment plant, influent, effluent, primary sources, probably washing of textiles. Some could be dietary. There's some types of PFAS, least toxic compounds used in pharmaceuticals, and maybe it's in fish and other food. That's something we need to do more research on. Again, total organic fluorine analysis, you need this to identify the presence of these complex mixtures of ultra shorts, which you need to look at in more detail. Risk in general, influent, effluent, it's driven by excess fluorine, but especially your terminal PFAS compounds in the effluent we see, because any precursor compounds are mostly broken down. Excess fluorine, there may be more there, but it's less toxic. But it could actually, in the wastewater treatment plant effluent, it is the excess fluorine PFAS that appear to be driving risk. Again, we're assuming that it's being conservative in its toxicity. Overall, the concentrations are really low. So they're low to moderate risk, moderate toxicity in comparison to drinking water action levels. If one question to come up is using wastewater effluent for irrigation, probably low to moderate risk to underlying drinking water aquifers. It depends on the irrigation method, hydrogeology of the site and such, more of a site-by-site call, but just overall for domestic wastewater, concentrations are already low to begin with. And it's mostly these shorter chain, less toxic, but still concerned compounds. Potential uptake into food crops, we need to do additional research on that using effluent for irrigation. Again, we think the risk for domestic plants would be, low to moderate. If the concentrations are low, we have a field study set up for next year. As far as discharge of effluent to aquatic habitat, surface water bodies would well below any published toxicity action levels for aquatic organisms, acute or chronic toxicity to the organisms themselves. Could there be some bioaccumulation in the aquatic food chain? Maybe. I'm not really concerned about that in a marine environment where a lot of the effluent gets discharged to in Hawaii. It's more of a focus from research we've read and talking to people who focus on filter feeders like clams, really absorb a lot of stuff in the wastewater, something to be concerned about. And freshwater fish, where you have wastewater treatment plants discharging into rivers where you're getting less mixing and dilution, especially if some of these wastewater pollutant plants are getting lots of landfill leachate, which the ones here aren't. We'll talk about in a minute. That could pose a problem for uptake and accumulation in fish and aquatic organisms. Then the concentration of toxicity of PFAS in wastewater from operations that use PFAS-based media, including operations where, say, they're using AFFF or something and it's getting flushed through the wastewater treatment system. That's a different story. The risk could be a lot higher for those. That's a different research. About the biosolids and compost in wastewater treatment plants, primary sources include washing of textiles again, especially here in Hawaii, we have lots of hotels and such, so it could be concerns about, especially the older types of tablecloths and bed linens, carpet, water stain resistant clothing by the main sources of PFAS in the biosolids and in the compost. TOPS processing, really important, especially for biosolids. I'd still do it on compost anywhere because that's your main risk is going to be driven by these secondary terminal PFAS compounds. These metabolites, precursors bound up in the biosolids. So the precursors, at least in the one sample we tested, seem to be getting oxidized in curing a compost. So they're the risk switches back to your primary terminal PFAS. And then still low toxicity in comparison to risk-based soil action levels. It's barely above 1 in most cases, not a big risk for even residential exposure. One point this question came up is that it's safe to dispose of biosolids at landfills. Well, definitely for the domestic biosolids, well below any action levels that would pose risk to landfill workers or people dropping off garbage or municipal waste periodically. And it's not going to add at all significantly to the PFAS burden in the landfill leachate. That's already coming from all the disposed municipal waste in the landfill. So not an issue there. Risk posed by uptake of PFAS in the produce grown in fresh biosolids is still likely low to moderate. Again, there's not that much there to begin with. Toxicities aren't that high, but that requires some additional research. I know some people are looking into this. One thought was that, remember, the biosolids themselves are mostly effluent, 85% moisture, or even higher in some cases. So probably it's when you first apply biosolids or to a field, even without making compost out of it, it's these short chain and ultra short compounds getting taken up pretty quickly into your first crops, released out to groundwater, and what's left is your less mobile compounds. It's probably initially it could be a problem, but aged biosolids in ag fields, I haven't looked into it, but the risk should be definitely a lot lower, either from uptake into plants or leaching into groundwater, but requires more research. PFAS and landfill leachate. Primary sources, again, textiles, carpet, furniture, treated paper, and such, a big mixture. Total organic fluorine analysis, you can use this to identify the presence of these complex mixtures of ultra-short compounds. There's still a lot of excess organic fluorine in the water. So, it's a moderate contribution to potential health risks. Overall, much higher risk and toxicity in comparison to wastewater treatment plants. Just comparing this to drinking water action levels again, so hypothetical risk of drinking water aquifers under leaky landfills. Again, the risk can be driven by primary terminal PFAS. A lot of excess fluorine in the water, but it's the primary PFAS that are going to drive risk because they're more toxic. There's not much precursor left. Threat to underlying aquifers in landfills. If it's a permit required, with engineered controls, landfill, it's intentional to prevent leakage of leachate from the landfills. The risk should be really low to underlying drinking water aquifers. We've been worried about leaking landfills for decades, not even related to PFAS, just all the other stuff that could be in landfills. So if you're following the engineered controls and those are working in place, should be no significant risk to underlying aquifers, but it's just something you have to watch. We have a lot of retired unlined landfills in Hawaii. They're mostly really small before we had the larger landfills put in place, but if they're near a drinking water aquifer, something we need to look at, then most of them have monitoring wells, so we need to add PFAS to the list of compounds monitored for these landfills, see if there's a potential issue. Question came up about disposal of landfill leachate at wastewater treatment plants, to get municipal wastewater treatment plants. So one thing they've done in Hawaii here is they limit the volume of landfill leachate that can be disposed of at a municipal wastewater treatment plant to it's around 0.1% from the landfills I've talked to, the wastewater treatment plants I've talked to, a total daily influent. So you're not going to even see any kind of bump in the total PFAS burden in the influent from this, or in the toxicity for the total PFAS going through the landfill. So you limit your daily disposal volume of leachate. It's not going to be an issue for the landfills, for the wastewater treatment plant. PFAS impacted soil. Pre-2005, AFFF is dominated by PFOS, driven by primary terminal PFAS. If you're not running tops on it, you're still capturing most of the risk. Anything after 2005, though, the precursor-based PFAS in your AFFF, you're going to miss it if you're not running tops on it. The risk is definitely driven by the secondary terminal PFAS and breakdown of those precursor compounds. Potential high toxicity, indirect exposure, even just compared to risk-based action levels and compared to, say, biosolids and such. Definitely something to look at. Ongoing leaching of PFAS from AFFF contaminated soil could be a long-term threat to underlying groundwater. That's why a lot of these airport sites now, where they have fire training areas, they're going in and digging up these old fire training pits. But just be sure to run tops on it in your confirmation samples. PFAS, AFFF-impacted groundwater impacts, again, pre-2005, AFFF dominated by primary terminal PFAS compounds like PFOS, hexasulfonate. Post-2005, it's going to be most of these short chain compounds, the butanoate, pentanoate, hexanoate. Could be a definitely potential high toxicity in comparison to our drinking water action levels. It's definitely beyond MCLs. The risk is driven by primary terminal PFAS in the source area. As you move in downgrading areas, though, in theory, if there are a lot of ultra shorts there, which appear to be the case, it could be these ultra short compounds driving potential risk if they're above a level of concern. So comparison of data to currently promulgated MCLs, and for long chain PFAS compounds, you just have PFOS, PFOA, and hexasulfonate. May not be adequate for groundwater impacted by some of the more recent releases of PFAS, of AFFF. Again, in the downgrading areas of Plume, our data is telling us we need to go out, and if we find any sites, drinking water wells impacted by AFFF or in that vicinity, we should be testing for total organic fluorine and getting the lab to report individual ultra short compounds to the extent possible. So low to potentially high acute to chronic toxicity if this groundwater got into surface water bodies, especially where we're not getting much circulation, either natural discharge or if you're dewatering a site because you're putting in underground utilities or something, you have to be really careful with that. Definitely in the source areas, you're way above acute toxicity action levels, even for aquatic organisms. Bioaccumulation in the aquatic food chain, discharges to wastewater or to surface water bodies. Again, more research needs to be done on this. Main concern from what we've seen is filter feeders like clams, and again, it's more a problem for rivers and impacts to freshwater fish where you're not getting a lot of mixing of your wastewater, or as your, in this case, AFFF contaminated groundwater as it goes into an aquatic habitat. So I end up here with looking at plumes, and we've been talking about for several years with some of our hydrogeology colleagues. So in this graph here, just imagine your source area if you dumped all the types of PFAS now we're focusing on, so our 24 different kinds of PFAS, including a few ultra shorts. Then what happens is the groundwater moves through this contaminated soil and migrates away from the source area. In the near source area, you'll still get, you'll get the shorter chain compounds, the more mobile compounds will show up. But the longer chain compounds, you're getting 8, 9, 10 or more carbons and fluorinated compounds, just not that mobile. They're going to stay in the source area. As you get down to kind of mid-plume areas, now you're starting to lose the less mobile, longer chain compounds, even shorter chain compounds also, mostly the carboxylates, short chain carboxylates. But this hexasulfonate seems to show up everywhere. So I think it's a lot more mobile than the absorption coefficient published would suggest. About the leading edge of the plume, then this is where at least we'd predict. We haven't looked for it. We don't have any big plumes to look at that we found yet, but try checking this in the distal downgrading edges of your, if you have any AFFF-impacted groundwater, especially. Look for these ultra short compounds. Have the lab test individual ultra shorts, trifluoroacetate, the ethanoate, or the propanoate, and also test for total organic fluorine and see if you find a lot of excess organic fluorine in the water. The theory would predict you would, and our data predict you would, but I haven't seen any data for it yet. That comes up into an issue, again, if you're treating groundwater or it's a source of drinking water for PFAS, if you're just running it through activated carbon, some of the remediation folks we've talked to have been pointing out, the activated carbon, it's going to capture most of the short chain compounds, your long chain compounds, but if these ultra short compounds are present, they're going to go straight through it. Again, less toxic, but still toxicity between naphthalene, benzene, that's such obviously something to worry about or to look into. So that's going to require some additional research and understand the toxicity when you get these really complex mixtures. So that was it. It's a summary then of our 4 years now research on PFAS. Hopefully we compiled a lot of ideas and information from our colleagues on the US mainland and elsewhere. We talked to a lot of people in Japan and China and Australia and in Europe. I think it's a good start. We're still doing additional research on, we're going to go out next year, collect some additional samples from wastewater treatment plants, and especially just to reconfirm what we're finding there. We think we see. We're doing a field study of uptake of PFAS into food crops. I'm actually heading off to China in 2 weeks from date of this presentation. Spend some time there assisting with research on PFAS, uptake into food crops and other issues. Working with some various groups on leaching of PFAS from soil and biosolids. So we use SPLP, synthetic precipitation leaching procedure, batch tests to look at standard, especially organic compounds, see how mobile they are. That doesn't work for PFAS that much because if there are any precursor compounds in that soil or biosolids, they're going to break down into these terminal PFAS in the SPL batch test solution. Really confounds the data. So the way to look at PFAS leaching, assess it, is through these soil column tests. It's method 1314. So we're working with some groups in the US and in Canada actually to come up with a cheaper and modified way to do soil column tests for PFAS contaminated media. In this case, you see in the picture on the left, you just fill a column up with your soil sample or whatever you're testing, compost, and then push water through it. You pump water through it. The volume of water you pump through it is equal, at least by Hawaii standards, to about 2 years of irrigation. So imagine you're irrigating a field or something, or just rainfall. Then you test the leachate coming out of the top. The standard lab method calls for testing the leachate 9 times, and there they're looking at breakthrough and such. Really, from a risk standpoint, we only need to test it twice. It's going to save a lot on money. We want to know what the mean concentration of PFAS is in the first flush, say the first couple of months of irrigation or rainfall through this soil. So we'll test it after about 5% or 10% of the water's been pushed through the total. It's going to be pushed through. Then you finish running the test and test the leachate again, and that gives you the long-term average concentration of PFAS. So we'll hopefully have some guidance set on that next year. Also looking at bioavailability of PFAS in soil and biosolids. We've been talking to some groups in Australia we've worked with in the past on that. PFAS vapor intrusion risk, of course, has to come up if these ultra-short compounds are out there. I recently talked to a chemist very familiar with AFFF-impacted groundwater and such, and by their thought was that in your ultra-short compounds there, the Henry's constant is just too low. They're not going to be that volatile, so they're predicting you're not going to see much in the term of vapors, at least from AFFF-impacted groundwater. That'll be useful to test. Any edits, suggestions, always welcome to study we're working on, our PFAS guidance. This time though, you're going to have to send them to John Jacob. He's our new state toxicologist. You see his email there. I'll explain why in a minute. I like these quotes. George Box, all models are wrong, but some are useful. So is our total PFAS approach absolutely correct and exact? Well, no, but we know it's wrong. It's not exact, but it's definitely useful. It's better than ignoring, these precursor compounds, all excess organic fluorine stuff. That's why I like the second quote there, John Maynard Keyes. It's better to be roughly right than precisely wrong. So that's where we are now. I think in the past, when we first started, we weren't looking at precursors. We weren't looking at all these ultra shorts, organic fluorine, excess organic fluorine. But now we are. So we're in the right path. We didn't get any questions. We were running out of time, obviously, during the original webinar. But I think we covered most of the ideas we've been talking about to our colleagues, PFAS colleagues elsewhere. Last couple of years, I'm sure there's a lot of work to be done. News on my part. So after 20 plus years with the Department of Health, I'm actually retiring into the end of November, heading off to China. I've been working there off and on for the last several years. Actually used to live there a long time ago. Been working with them on PFAS research. I'll be staying in touch with the group here in Hawaii and other folks. I still plan to stay active in environmental work for the foreseeable future. So my work email is not going to work anymore after November 30th, 2024. So you see my new work email at the bottom, tosbrewer@yahoo.com, a little nod to Theory of Sampling colleagues at the bottom. So keep in touch. It's been a great 20, 30 years now, and I look forward to the next few years. Thanks.